What is a cell? Where do they come from? How do we study cells? How did we work it all out in the first place? And why does this key idea of this fascinating topic still underpin the whole of the science of biology? Amino Man and Gluco Girl might be able to help you understand it just a little better.
Welcome to this first ever video explainer of a series intended to help you make some sense of the beast of knowledge that is A-level biology. This short revision explainer covers the first part of the topic cell structure, which in the AQA course is topic 2. All of the notes and extra study resources can be found on Van Dyke Upper School Show My Homework, under the teacher filter of Mr. Chantrell, which matches up with the work covered in this video.
If you're studying biology, you have to know about the cell theory. 300 years in the making, some of the titans of biology have gathered evidence towards it, or used elements of it in their own thinking. The cell theory is probably one of the most important ideas in the whole of biology, and practically everything else you learn in this subject links back to it. But what is the cell theory? In this video, we will try and give you a clear cell theory definition, explain key dates in the history of it, and explain why it's so important to understand. We will then look at how that important info was gathered together, mostly thanks to the superb visualizing tool of the very small, the microscope.
Okay, on to the key facts about the cell theory. It has two main parts. Number 1. All living things are made up of cells. That means literally everything, from bacteria to bees, humans to hyacinths. Number 2. All cells come from pre-existing cells created through the process of cell division. This is an idea, of course, that you have come across at GCSE through the study of mitosis.
As time has gone on, more components have been added to the theory. It's useful to be aware of them, particularly for later units that we cover, especially in year 13. Next, number 3. Energy flow occurs within cells, particularly important in chemical reactions such as respiration and photosynthesis. Number 4. Hereditary information is passed from cell to cell, the inheritance of DNA molecules donated from the parent cells to the offspring cells. Number 5. All cells have the same basic chemical composition. One of the great truths about the cell is that essentially all living things have the same chemical toolkit. The only real difference is what those living things tend to do when they combine the use of those tools in different ways. This is really what makes cell types and species different from each other, and it is the key idea that underpins the other side of the course about biological molecules and enzymes.
Okay, some brief info now about the history of the theory itself. The first pioneering work was carried out by the Dutch draper Anton van Leeuwenhoek, who in the 17th century through his work on cloth became Europe's expert on all things to do with the new device that was the microscope. If ever somebody could be said to be obsessed by this new technology born out of the glass revolution of the Renaissance, it was him, and he took to experimenting on his own body, eventually discovering a whole zoo of microbes such as bacteria, and even his own sperm. Off the back of his work came Robert Hooke, who with slightly inferior equipment and bigger objects to look at in plant matter, was the first person to coin the term cell to describe what he saw inside a piece of cork, named after the prayer cells that often found in monasteries.
Other scientists contributed much over the next few hundred years, but the full theory and its ultimate affirmation had to wait until the late 19th century, especially the work of Robert Brown, Schleiben and Schwann, and the great Louis Pasteur, the great practical biologist, who with his brilliantly designed swan neck flask experiment proved that life couldn't arise from nothing, which you may have learnt all about during your own history GCSE. His revelations eventually killed off the rival theory of spontaneous generation, leaving the cell theory as the only theory that could adequately explain the observed evidence, and eventually came to be the foundation for what biologists could see of life at the smallest level.
The next 150 years allowed even more evidence to be gathered that backs the cell theory to be in the main true, and as with all good theories, it is often the exceptions that prove the rule, such as viruses.
Next, let's talk about how we see inside cells. Technology has allowed us to peer deeper and deeper into living things. and some truly magical techniques have revealed objects of such incredibly small size, yet of such intricate beauty and complexity. The most important of all these devices is the light microscope, and until the last 70 years, that was all that was available. Most students are quite blasé about microscopes through their blurry and unfocused experience at GCSE, but just reflect on what an era-shifting piece of technology it truly is.
Remember, a tool is only ever as good as the craftsman that wields it, and at 15 or 16 years of age, students are never going to know enough to unlock its full potential. The first use of microscopes revealed a previously unknown world of objects previously unknown, and allowed scientists to begin to work and think on a level that was far smaller than anybody had ever appreciated. In fact, it is so useful that we can easily measure, even with school equipment, objects that are far beyond the range of the human eye. Light microscopes are cheap, but can be used on living matter without the need to kill it first. They are, however, limited by their wave of choice. Light waves used to illuminate their objects. Their limit is to distinct points half the diameter of a wavelength of light apart. That is so often why research labs often use shorter wavelengths of M, such as blue light or even ultraviolet, in order to push this equipment to its operational maximum.
The most important info about microscopes is the famous IAM equation linking image size, actual size and magnification. Like with any formula, it is best laid out as a formula triangle, and as a tip, it is best to learn the order in a way that you could reproduce in an exam. I suggest using IAM anti-clockwise and deploy the equation as required by a specific question. The other major focus of microscopy should be the scale that is usually used to measure microscopic objects, handily referred to as micrometer, or sometimes micron for short. This measurement is 1/1000th of a millimeter, or 1/1,000,000th of a meter, commonly described as 1 × 10⁻⁶ m, 0.00001 of a meter. Convert all units into microns before you begin a calculation to reduce levels of heartbreak in exams later.
The final part of this video covers electron microscopy, the technique that more than any other has revolutionized the way we see not just whole cells, but also the constituent parts or fractions of those cells. They're organelles that carry out the individual functions that cells are capable of carrying out.
Electron microscopes are both better and worse than light microscopes. Prohibitively expensive, they cost hundreds of thousands of pounds at the latest count, and therefore are usually only within the reach of corporations and higher education institutions. They are far from being tabletop machines, so are fairly inconvenient to use. They also have the major two downsides, firstly having to kill whatever it is you are having to look at, and then having to coat it in a material that allows it to interact with electrons like a fine layer of gold. At this point, it is best to imagine James Bond and the movie Goldfinger here. They also generate ionizing radiation as a byproduct of making beams of electrons required to make the object visible.
But when it works, it gives fantastical images of things that are so small, yet so detailed, that they are almost beyond comprehension. This is possible because electron waves, yes, electrons are waves as well as particles, speak to your physics teacher for a more detailed explanation, have a much smaller wavelength than light. Light wavelengths go from 700 to 400 nanometers, red and blue light respectively. Electron waves are only 1 nanometer, giving incredible resolving power when compared to a light microscope. Two basic kinds exist. A scanning microscope, commonly called an SEM, that shows the outside surface of objects in beautiful 3D, but things that are comparatively far apart, so they are said to have poor resolution. And the transmission electron microscope, or TEM for short, that can see images in cross-section and give the best resolution.
A final point worth mentioning about electron microscopy is that the technique continues to evolve and be improved. One improvement in particular has the potential to show molecules at work, not just organelles. This is known as cryo-electron microscopy, or cryo-EM for short, and for many years was seen as a wacky alternative to other techniques for decades. This was because the resulting images were little more than blobs on the imaging screen, hence the rude description of blobology by its detractors. but over time, scientists refined cryo-EM to the point that it earned a Nobel Prize for Chemistry in 2017. There has been a perfect storm of improvements in camera technology, image processing, reduced cost of parts as well as increased computing power, which has utterly transformed the field and is starting to show researchers exactly what is actually going on in cells and so paving the way for the development of better drugs for infectious disease.
These include those that are challenging to treat, such as flu, HIV, coronavirus, and many other diseases associated with dementia such as Alzheimer's and Parkinson's disease. Using cryo-EM has been likened to seeing how all the different parts of an orchestra play individually to create a piece of music, rather than just looking at the sum total of all the noise of all the instruments playing together. Truly revolutionary.
As we are novices when it comes to making these kinds of videos, we would really would appreciate your feedback in the comments section of this channel or by email, especially if you think a different voice is required to present it. This video is intended to support the first look learning resources used in class. Having a complete set of notes that you can add to with your own work from extra study is the most crucial part of your A-level studies. There are no shortcuts in biology. You must become a biologist in order to maximize your potential. Some more links can be found at the very end of the video. Signing off. I will hopefully catch you young guys and gals later in another video explainer.